All right, so we're in chapter 6 here. We finished up our control of bacteria talking about antibiotics. And this is a brilliant tool for killing bacteria. These other kinds of physical methods, things like heating stuff up, using UV light, using radiation, pasteurization versus sterilization, using phenol. These are fairly nasty things that you can't really do to people much, internally anyway. But antibiotics are a brilliant tool. I mean, this is why the development, the discovery of antibiotics at the early part of the 20th century is a revolutionary part of medical health care. Because prior to this, if you had a bacterial infection, about the only thing we could do is say, hold on and hope your immune system can fight it, or look at it and go, dude, the bacterial infection is here. We got to prevent it from spreading, and so we're going to take your leg off at the knee. And pretty much if you had a compound fracture in which the bone is exposed and there's tons of infection going on there, now what they would do is they would set it and close everything up and put you on massive antibiotics. 100 years ago, they'd look at that and go, Hoss, we're going to have to take that leg. And so, and then you'd be lucky to survive that. So antibiotics, are really a serious game changer. But as we were looking, antibiotic resistance is a real thing. And you generate antibiotic resistance by either generating an enzyme that breaks down the antibiotic. That's the standard thing for all the illin type antibiotics, ampicillin, penicillin, amoxicillin. Or you have a mutation in whatever protein system that the antibiotic is interfering with. Just as an example, streptomycin interacts with the ribosome in terms of protein translation. And if you tweak a single amino acid in a ribosomal protein, all of a sudden strep doesn't bind anymore, and you're now resistant. So because it is either an enzyme or a structural protein that can confer resistance, that is transferable in terms of genetics. The progeny will have it, but you also can have what we call horizontal gene transfer, where a little bit of the DNA goes from one bacterium to another. And that's what we're going to look at. So some of the basics that we've already gone through is the genome, the chromosome of a bacterium, is a circular chromosome. And it is in the nucleoid, not the nucleus. We've got 46 chromosomes. Bacteria have a single chromosome. This is the way they work. Additionally, bacteria can have these things we call plasmids, which are circular DNAs. They're a lot smaller than the chromosome, so you're not going to confuse them with the chromosome, but they're replicated along with the bacteria. Actually, some plasmids can replicate multiple times within a bacteria, so you can have what's known as a relaxed plasmid, in that you might have a couple of hundred copies of it within a single cell. Other plasmids are referred to as stringent, and they basically replicate along with the chromosome, so you have one per cell. We use them in cloning all the time. I mean, when we start talking about cloning and recombinant DNA, we're using plasmids and letting the bacteria copy it. So, additionally, because of the chromosomal organization, bacteria are what we would think of in the genetics world as haploid, meaning that you don't have a chromosome and a gene copy from mom and a chromosome and a gene copy from dad, like we do, diploid, except in our gametes. They have a single copy, and that makes it a lot more sensitive to mutation. I'll tell you that, is that you can pick up a mutation in one of your genes that makes it non-functional, but as long as you have the other functional one, you're probably going to be okay, at least for a while. But in bacteria, they pick up a mutation, that's it. They don't have a backup copy. So binary fission is how they're separating. And the whole advantage behind what we call mitosis is that it provides for the possibility of having some changes in the DNA, and actually, that's meiosis, but regardless, they don't go through mitosis in the same way. They are just simply replicating the DNA. In this case, with being asexual, meiosis is what generates the rearrangements and the recombining of chromosomes. And so that's why your brothers and sisters don't look exactly like you unless you are, in fact, specific zygotic twins, because you've got slight recombinations and rearrangements of chromosomes. Bacteria don't do that. So your bacteria grandchild looks exactly like their bacteria grandfather, grandmother, and great-grandmother. And additionally, we've got the replication or the transfer of genetic material in a vertical fashion, which is what we do. So you have parents generating offspring. But bacteria also can carry out what's called horizontal gene transfer, in which you've got DNA that is in one cell, can be moved around by a lot of different mechanisms, and picked up by another cell. Two guys are not related in terms of I begat you, it's just that you happen to be near me. And so this doesn't happen with us either. Still, the central dogma is the same. DNA is replicated, DNA is transcribed to RNA, RNA is translated to proteins. That doesn't change. And in fact, the genetic code is identical. So it means that you can take a bacterial gene and express it in a eukaryote, and you can take a eukaryotic gene and express it in bacteria. I was just thinking about this earlier today, that when you go, when you start to plant corn and soybeans around here, pretty much all of it is genetically modified, and all the corn and all the soybeans are carrying a bacterial gene that codes for a protein that chews up the little guts of chewing insects. And you have to have some differences in the control system, but once that RNA is produced, the plant cell is able to translate it just the same way the bacteria does. So what this means is that you don't get recombination, you don't have scrambling of chromosomes. Anything that's going to happen and change is all because of DNA mistakes. So this is a list of terms here. Hopefully you've got some of these before. Genetics is what we're talking about. A gene we can define in a couple of different ways, but the best definition is a piece of DNA that codes for either a trait, a protein. The better, more generic way is a piece of the chromosome that can be transcribed, produces an RNA. Because not all RNAs are being translated, but in order for a gene to actually do something, you've got to be able to build the RNA, make the RNA out of it. So the genetic material, primarily all cells and lots of viruses are using DNA. There are, in fact, no viruses like the coronavirus that use nothing but RNA. The genome is what we refer to when we're talking about all of the information that is held on those chromosomes. The nucleotides, of course, those are the monomers that make up DNA and RNA. The sequence is critical because that's how we can begin to think about the proteins that could be generated from a given nucleic acid sequence. Replication, transcription, we've already talked a little bit about that. Producing RNA. Translation is going from RNA to protein. And a mutation is anything that changes the nucleotide sequence. So some basic terms here that we will be using and kind of referring to as we continue on. So the understanding that DNA was or is the genetic material didn't happen overnight. And if we think about what we know of experimental systems and how you're operating with this, and what we know about our biopolymers, let's think about this for a second. in our list of biopolymers, we have carbohydrates, and they pretty much are just carrying a net negative charge. They make good pudding, but they don't have a lot of interesting structure. Fats, lipids, they make, these fatty acid chains, and they're nonpolar, but again, they don't form up into interesting structures. Nucleic acids have the 4 different nucleotides, and they can fold up and do base pairing to generate something with some structure, but the structure is somewhat limited. When you get to proteins, we got 20 amino acids, and the possible combinations of these 20 amino acids is now really, really big. And we've got amino acids that are positively charged, amino acids that are negatively charged. We got polar, nonpolar. We have the ability to form all these amazing structures. Now, they're held together by weak-ass bonds, but still, they can form these really complex structures that allow them to carry out enzymatic activity, to bind stuff. They can do things. So, at the early stages of thinking about genetics, Mendel was one of the first guys to actually begin demonstrating the idea of a trait and an allele, that is, a specific sort of a form or a flavor of a trait, curly versus straight, or blonde hair versus black hair, blue eyes versus red eyes, whatever. The next step is, all right, how are these traits being transferred and moved around? And back in, actually before World War I, a guy at Caltech by the name of Morgan was working on fruit flies, and flies have chromosomes that are pretty easy to actually see in the salivary glands. You have to squash them from a microscope, but you can see them. And they got these bumps and ridges and banding patterns that are invariant, and so you can actually keep track of them. And he was able to posit and recognize what he called the chromosomal basis of inheritance. So this is the first step to say, all right, inheritance is happening because chromosomes are being moved from one cell to another. When you look at chromosomes, okay, they're made up of this nucleic acid stuff. Nucleic acids didn't seem to be that much more interesting than starch. I mean, it's just a real big negative charge. They don't have much in the way of enzymatic activity. They really don't fold up into dramatically interesting structures. Probably the genetic information is proteins that are held on the chromosome, and that's how they're being moved around. So this chromosomal basis of inheritance, big paper, big deal, but really the DNA is a scaffold for bringing different proteins around. I mean, that ain't right, but that's what would be an obvious thought. So you then have to go forward another 20 or 30 years to this really critical experiment that is demonstrating that DNA is, in fact, the genetic material, not the proteins associated with it. And this is an experiment by a guy named Oswald Avery. And what he was doing was developing the tools that allow bacteria to take up DNA. So he had a selection of pneumococcal bacteria that had 2 different mutations. Essentially, the type R is non-virulent. You inject it into a little mouse, the mouse doesn't care. Actually, what it is, it's a mutation that doesn't have the lipopolysaccharide around it. So it doesn't have the candy coating. It is not toxic to the bacteria or to the mouse. The mouse doesn't care. You inject it, the mouse walks away. He then had another bacterial type, we refer to as type S. Rough meant it was rough, or R meant it was rough because it didn't have the candy coating. It actually was looking bumpy. S was a smooth phenotype. It has the candy coating, the lipopolysaccharide on it. And you inject that into the rat, and he dies. So, when you look at the blood, you can find that the virulent type is present there. Now, when you take the S type and you heat kill it and inject it, the rat's probably not thrilled with it, but the bacteria can't replicate, they can't divide, and he survives. But what he found was, if you took the live non-pathogenic R-type and mixed them with the heat-killed S-type, you would transform the R-type into the S-type. And so he coined this term, transformation. And the next question is, what is the transforming factor? Bridges would have told him, or Morgan would have told him, dude, it's protein. What he did was begin cracking open the heat-killed guys and fractionating different parts. He put the protein in with the R cells. He put the lipid component in. Nothing happens. He puts the nucleic acid component in, and bada bing, his experiment works. He has determined that nucleic acids, DNA specifically, are in fact the transforming factor. This is a pretty amazing thing. And back in the 40s when he was doing this, we have pretty good proteases that can break down protein. So you can take a soup of dead bacteria, split them open, incubate it with protease, which will remove all the protein, incubate it with RNases to remove all the RNA, lipases will remove all the different lipids, and so you can be left with a reasonably clean DNA prep. And it turns out that works. So we do this in the molecular biology lab all the time, transformation. And so now we know that DNA is in fact the transforming factor. DNA is the information. So the next thing that starts to happen as analytical chemistry starts to get a little bit better, begin looking at what is nucleic acid. We have the 4 bases, adenine, guanine, cytosine, thymine. And Chargaff, who may have actually been at U of I, because some of this stuff was being done over in Urbana, he was able, by careful analysis, to demonstrate and determine that the amount of adenine would always be equivalent to the amount of thymine. The amount of guanine is always equivalent to the amount of cytosine. Now, he didn't know what this means, but it's a pretty obvious thing that starts to lay out. It's like, damn. So it's not like looking at a protein where you hydrolyze it and look at the number of amino acids. I mean, with all 20, you don't see any kind of rules of, if you've got this amino acid, you're always going to have that amino acid. Nah, it's not nearly that clear. But for DNA, this is an interesting kind of phenomenon. So people continue working on this problem. And another brilliant experiment that confirmed the Avery experiment, the DNA as a transforming factor, is a classic experiment known as the Hershey-Chase experiment. And this is done at Cold Spring Harbor Labs out in Long Island. We have bacteriophage. These are viruses that infect bacteria. And you could grow the bacteria in a media that contains radioactive tracers. So one thing you know is this is past 1940, past World War II, because we finally have the ability to generate radioactive tracers. That's a really critical thing. You label up proteins using sulfur-35, which is a isotope of sulfur. We know that sulfur is not going to get incorporated into the nucleic acid, and sulfur is a part of the amino acid methionine and the amino acid cysteine. So you feed the bacteria radioactive sulfate, and they incorporate that into their proteins. So you can label the proteins, and then you can take phosphorus, radioactive phosphorus, and feed that into the bacteria, and they will incorporate phosphorus into the nucleic acids. So you grow the bacteria virus on this mix, and they end up with this label in which you've got the protein is S35 labeled, the DNA is P32 labeled, and they then do the experiment of infecting bacteria with bacteriophage. And really all you do is you take a little bit of the phage solution, either spot it onto a plate or shoot a little bit into your bacteria culture, wait a few minutes, and the bacteria, the phage, infect the bacteria. The virus is, as we now know, injecting its DNA into the cell. So they then took, after letting it sit for 15, 20 minutes, they took the bacteria and the phage, and then they threw it into a blender and whizzed it up really hard. And what that's doing is knocking the bacteria phage off the cell surface. And they were able to identify that with the protein label, all of the protein is floating out in the juice. None of the labeled proteins getting into the cell. With the P32, when they do all the blending and then spinning to the bacteria, they find all the radioactive P32 is in the cell. That, again, is the final bit of information that says DNA is what's transferring information, not protein at all. A really pretty big deal here. Nobel Prize winning. So then people really began studying DNA. This is a X-ray diffraction pattern generated by Franklin and Wilkins, who didn't receive the Nobel Prize, but should have, from the structure. Now, I don't interpret or begin to interpret X-ray diffraction, but from this photo, people were able to get an understanding about how the DNA molecule was being arranged. That it is a helix, that there is very specific constraints to the width and the way the helix is arranged, and there is a repetition that is like the spiral. And so from this information, we get the double-stranded DNA molecule that we know. That is what's known as the Watson and Crick model. So I think Jim Watson is still alive. He's a very old man and a fairly nasty guy and a bit of an asshole. But they did have this amazing moment. Initially, when they were looking at the structure, they recognized that the nucleotides, or the bases, were the thing that were different. They had Chargaff's rules, and so they knew that the A base pair, or the A interacted with T somehow. But because the phosphorus backbone, the phosphodiester backbone, is always the same, they figured the bases had to be sticking out. And so if you could, develop a time machine and go back to 1953 or something and whisper in their ear, dude, the bases go inside, they might have to have given you the Nobel Prize as well. So it takes them a little while to figure out that, all right, this isn't working. What if we turn it around? Ooh, adenine can base pair through hydrogen bonds with thymine. Guanine can base pair with cytosine through hydrogen bonds. Dude, this works. And this helical structure is what we now know is the basic structure of a double-stranded DNA molecule. So if you think about helices, and I often do, imagine a spiral staircase that doesn't have any handrails. So it's actually not OSHA certified. It's a little dangerous. But you're walking up a spiral staircase. What you're walking on is the hydrogen bonds between base pairs. And the stringers, as they're called, or the edges of the stair, the spiral stair, that's the phosphate and sugar backbone. And if you think about this, your feet are between the strands. And your feet are in what is referred to as the minor groove. But then because this thing is a helical structure, your body is in between two parts of the stairs. So you're walking around, your body is here, you're going to be actually walking up the stairs, and you're going to be moving around. Your body is filling up what's known as the major groove. And so there are proteins that interact with both the minor groove and the major groove. And this is a fairly remarkable discovery now, that we've got genetic material that we can actually begin to use and analyze. So this model works in that they demonstrate that A is interacting with T, which means that A equals T, that G equals C. That ratio is fixed in this species, and that makes sense. The angstrom X-ray crystallography has to do with the difference between the bases and the distance between the twists, that begins to make sense. Nothing is restricting the order of the bases. You can have any range of bases. The hydrogen bonding between the bases allows for it to open up so you can get proteins in, like RNA polymerase. And then their last statement, it has not escaped our notice the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Ha! Yeah. So, 1953. We now know by a bunch of different experiments that DNA is in fact coding for proteins. So this is an electron micrograph in which we lyse open bacteria and plop them onto a very thin film of like epoxy sheeting that's on a little grid that is like the size of the back end of your pencil eraser. And you put that into an electron microscope. And here is the cell that's been blown apart. This is the genome spraying out. Kind of like if you imagine a 50-yard long ball of yarn and you stretch it out. You take the two ends, tie them together, so it's a loop. But, once you start balling it all together, it's just a mess. That's what the genome is like. And then you can find these individual circles of much smaller DNA molecules. These are the plasmids. Not all bacteria carry plasmids, but most do. And so you've got the chromosomal DNA and then what we think of as the extrachromosomal circular plasmids. And just as a little point, you and I cannot carry or replicate extrachromosomal pieces of DNA. The only way we can replicate new DNA is if it integrates into our chromosome. And so you can inject or cause cells to take up plasmids, eukaryotic cells, but as they divide, they just start kind of divvying up that plasmid DNA, and it just is split up fairly quickly until there's not much left. But that's because we cannot maintain them and replicate them. But bacteria can. So this leads us then to some of the mechanisms behind how we can replicate DNA. And this is just as a little blurb here, but you are going to take the double-stranded DNA, open it apart, and DNA polymerase is adding new nucleotides. And so this is referred to as semi-conservative replication because every new strand has one parental strand and one new strand. So the fork runs from one side to the other, and that's how you begin to replicate DNA. So in chromosomes that are linear, like ours, you actually have multiple spots of origins of replication, as they're called, and you're just zipping off at multiple spots of this bubble. With bacteria, you have a single origin of replication, both on the plasmid and the genome. And what that means is you start here, and you start replicating, and as you do that, you have replication forks on either side, and you begin actually to make something that looks like the Greek letter of phi, or phi, the circle with the line across it. And when you get to the end, you have two DNA molecules now, two double-stranded DNA molecules. So this is what we refer to as vertical gene transmission. DNA replication that is really just like mitosis in eukaryotes. So the bacteria is replicating its genome. That's easy. Now, the other way we can think about this, let's see. We'll get that a little bit later. We're going to talk about horizontal gene transfer later. So once you have a gene, or once you have the DNA, in order to actually make an RNA from it, we require RNA polymerase. This is the enzyme that actually opens up the double-stranded DNA and it reads the bottom strand and generates a complementary copy to it. And so we refer to this as the template strand, and this is the non-template. Another term is this is the sense strand, because this is actually the same copy, except T is now U, but it's kind of irrelevant, as the RNA produced. And this is the complement to it, the base pair complement. So once we've got the RNA, we can then begin to translate that RNA into proteins. And we got a little bit more to say about that. Let's see here. All right, just to go through it a little bit more. And I could draw it, but we'll use this figure here. The material, or the organelle, is the ribosome that takes the RNA and turns that into protein. And I'm not sure where you get this information from. You haven't had genetics necessarily. We read the DNA 3 nucleotides at a time. And tRNAs are coupled to an amino acid, and they are interacting with the RNA strand. And the ribosome moves down 3 nucleotides at a time, and we begin to generate a peptide bond. The enzyme is actually integral to the ribosome, and you begin to link together the amino acids. So you've got methionine here in this slot. The next amino acid comes in. You make a peptide bond here, pop the methionine off of its tRNA. That tRNA just floats away, and this whole thing shifts over, and this tRNA is now in this slot. So you just start working your way down from the 5' end of the RNA to the 3' end, making your way in and making a protein. So this is a very simplified way of thinking about translation, but it gets the job done here. So here is the bottom line then, and we're going to pause for the day. We've got DNA that can direct its own replication. DNA can also direct the production of a portion of it, and that is transcription. It makes an RNA copy. RNA is able to direct the production of proteins based on its sequence, and that is translation. And of course, the link is the RNA sequence is dependent on the DNA sequence. Consequently, any change you make to the DNA can affect protein. And that gets us to the nuts and bolts of mutation, and we'll pick up on that on Friday.